So as I was saying, oh, someone intruded. I've even forgotten where I got to. So I'm sure I was talking about the DNA methylation, right? So when, let's say, a particular gene has its control region methylated, you need your control region, like the promoters and enhancers, to promote transcription. So if those sequences or if those structures are methylated, they are going to be inactive. Hence, transcription cannot occur. Okay, so but the unmethylated version of the gene will be expressed. Okay, so replication of methylated allele. So if the cell is replicating, okay, you realize just like a DNA replicates, it will unwind into two strands. So if you have like four DNA, so let's say four methylations on your DNA, and your DNA unwinds like two on each strand, you realize each of the daughter cells will have two methylations. So those, we call them hemimethylations. So your daughter cells will be hemimethylated, they won't be fully methylated. So as I said, because these structures are self-perpetuating, they will have to find a way to maintain, or like should I call it fix, or make it fully methylated, right? So replication does not affect the state of unmethylated allele. So you realize in this structure you had an allele like this, right? It had methyl group here, methyl group here. After replication, it will have, where is it? Oh, it's not showing well. Yes, so you have this allele with two methyl groups, right? But after replication, it now forms two daughter cells. Okay, it forms two daughter cells, each one with methyl groups instead of each one with single methyl groups instead of two methyl groups. So now the lost methyl groups has to be restored. They have to be restored, right? So this enzyme will come in, the methyl transferase, DNMTs. We have different types of DNMTs, and we will talk about them later on. But DNMTs will come in and methylate the demethylated regions, okay, to make them fully methylated. But for normal alleles, or for non-methylated alleles, these methyl transferases are not going to work on them, because they are not, like, they don't have, should I say, they don't have the history of being methylated. Okay, because previously these daughter cells were not methylated. Their parental structure weren't their parental structures weren't methylated. So the DNMT coming in, that's methylating this, is not going to affect this. Okay, that's what we mean by replication does not affect the state of unmethylated allele. Okay, so let's look at this, and we are going to look at the different types of DNMTs we have. How the methylation patterns are maintained as well. So the first one is a normal DNA with two methyl groups on each strand. Your DNA is going to replicate, and the daughter cell will have one. So there should be a way of maintaining the methyl group that's lost. So we have a maintenance DNMT called DNMT1. Okay, so DNMT1 is a maintenance methyl transferase. So DNMT1 will restore the lost methyl group, right? So what if it's not restored and it goes on to do a second round replication? The daughter cell might lose. So if the strand without the methyl group will form a daughter cell with no methyl transferases. Okay, but even though this is an unmethylated allele, it still has a history of being methylated in the past. So its ancestors were methylated. So this one will be affected by DNMT. So we can have de novo DNMTs that can methylate this into a fully methylated structure. So you can have two DNA molecules, each one both of them with no methyl groups, but the one with a history of being methylated in the past, or let's say it has ancestors that were methylated in the past, that one can be affected, or that one can serve as a substrate to these de novo methyl transferases, DNMT 3A and 3B. Okay, but the DNA structure that has no history of being methylated is not going to be affected by methyl transferases. So the takeaway from this picture is we have two types of DNMTs: maintenance metallotransferases and de novo metallotransferases. Maintenance is after replication, you form a hemi-methylated structure. Like one is missing, so the maintenance is going to just repair it, right? And the de novo is like synthesizing from scratch. So what if the hemi-methylated ones are not fixed and it goes on to replicate into daughter cells, and obviously one of the daughter cells from the hemi-methylated structures is not going to have methyl group. Okay, even though this structure is unmethylated, it has a history of being methylated in the past, so the metal transferases can work on it. Okay, so that's where de novo metal transferases come in. And the types of de novo metal transferases are DNMT 3A and 3B. Okay, so mammalian, they are saying mammalian DNMTs are very essential, and this is what they do. In mammals, we have DNMT 1, 2, 3A, 3B, and 3I. Right. So in 3B, when you lack DNMT 1, or if your DNMT 1s are not working, what are DNMT 1s? They are maintenances. So if your DNMT 1, they are maintenance metal transferases. So if your DNMT 1 is not working, it is very lethal to your embryo. Okay. For DNMT 2, there is no known obvious effect. For 3A and 3B, the de novo ones, they are very lethal for perinatal and also the embryonic the embryo as well. For 3I, there are no imprints. Okay, so the most essential ones are the 1, 3A, and 3B. So let's look at these self-perpetuating structures, the protein aggregates, and the things but modifications that causes epigenetics. Okay. So the ability of these proteins, let me read it all. Perpetuation depends on the ability of the proteins in heterochromatic region to remain bound to the region after replication, and also to recruit more protein subunits to sustain the complex. Okay, so just like the methylations on DNA, protein structures on the histone must also have the ability to remain bound, and also after replication, just like methylation, some they will be like half the number of proteins that were on the parental structure. So they should have the ability to recruit more proteins to sustain themselves. Okay, so just like this, these are the green ones here. The green structures here are the, let's call them the self-perpetuating structures, the proteins. So after replication, you realize instead of having 4 proteins, each one of the daughter chromosomes, or let me say the resulting chromosomes, will have 2 instead of 4. So the proteins should have the ability to recruit more. So this is what we call protein self-assembly. So it will now I don't know whether it's creating more proteins of itself or recruiting more. So it will restore its original number 4, right? But in euchromatin state, there will be no addition of protein, because initially there were no proteins on the parental structure. The protein responsible for such situation must have something called self-templating or self-assembling capacities to restore the original complex. So unlike the DNA methylation, that will require DNMT to come in and restore the methylation marks. For these ones, the protein themselves should have the ability to self-assemble. Okay, so they should have an ability called self-templating or self-assembling, right? And the 2 daughter chromatids will continue to be marked by the protein. Histone modification is responsible for epigenetic effects. I've mentioned this. So mostly the histones we modificate. But when I modificate the histones we modify. I think in lecture 4, the different histone proteins that take part in chromosome folding. Okay, we have one. Now one is actually a linker, so 2A, 2B, 3 and 4, right? So mostly the modifications are done on 3 and 4 on their tails. I think I read somewhere that the reason why it's mostly done on 2A actually also plays part, 2B also, but mostly they are on H3 and 4. The reason being their tails are more available and long, blah blah, visa. So you can also search about why most the histone modifications are on H3 and H4. So I think I've mentioned this. If a heterochromatin becomes acylated, heterochromatin is an inactive gene. Okay, if we acetylate a heterochromatin, it becomes active, because mostly acetylation in histones deals with activating, and the methylation mostly deals with deactivating or inactivation, right? So histone methylation can activate or repress gene expression. I think I've said this as well. So depending on the lysine residue you are methylating, you can either activate or repress. So as I said, if you are methylating K9, K27, you are going to repress gene expression. But if you are methylating K4 and say K14, you are going to activate. Okay. So histones also have their own methyl transferases called histone methyl transferases, and these are how to add the methyl groups. They do not add more than 3 groups. So a histone can be monomethylated, dimethylated, and trimethylated. Okay, so I think we will come across codes. You come these codes: H3K9ME1, H3K9ME2, H3K9. H3 is the histone 3, K9 is lysine 9, that's like the 9th residue, and Me is, that means that it's monomethylated. So Me2 there is dimethylated, and Me3 is trimethylated. Okay, so mostly methylations on lysine 9, as I said, deals with repression or gene inactivation, right? But if you are methylating like K4, that deals with activating the genes, or K14. I think you come across that one as well. So addition of methyl groups to histones can either activate, we've said this. So methylation of specific lysine residues creates binding site for other proteins. Okay, and this is what we'll be doing for mostly in this part of the video, looking at. So what you have to take home from these pages: when we methylate the DNA, especially creating heterochromatic regions, okay, on K9 and K27, we are mostly methylating to provide binding sites for protein structures, mostly HP1, cell group proteins, and polycomb proteins. Okay, that's when we are deactivating, or like called repressing gene expression. But when we are activating, we are causing binding site for the trithorax groups. Okay, we will take them one by one. There are sections dedicated to each one of them. So heterochromatin propagates from a nucleation event. Nucleation is like the first step, like whatever initiates it. That's what I understand by nucleation. But let's just see what they're talking about. An interphase nucleus contains both. I'm sure you know interphase. So during the cell cycle, we have the S, G1, G2, and the M, right? So the G1, S, and G2 phases are called the interphase states, or the interphase phases. Okay, and the M is like the prophase or then, and the M will contain a prophase, anaphase, then. Right. So the interphase, the S, G1, and G2. So if a cell is in the interphase state or in the interphase phase, its nucleus will contain both achromatin and heterochromatin. Okay, and condensation state of heterochromatin, blah 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 blah. Heterochromatin is generally innate. I'm sure we know this because it is inactive, right? So therefore, heterochromatin is going to remain condensed in the interphase, and it is transcriptionally repressed. It replicates later in the S phase. So mostly in replication, cell replication, the achromatic regions are replicated first, and the heterochromatic regions are replicated later in the S phase. Okay, and they are mostly localized at the nuclear periphery. I think this will be talked about as well. They are associated with the nuclear lamina and stuff. So centromeric, if you can recall the structure of a chromosome, our linear chromosome, you realize we have the centromere, telomeres, right? So mostly these regions are always like permanently heterochromatic. The centromere and the two telomeres, they are permanently heterochromatic. And for heterochromatic regions, we have two types. We have permanent regions and parts that can be, activated to achromatin and also inactivated to heterochromatin. But for the centromere and the telomeres, they are permanent heterochromatic regions. Okay. So the centromeric heterochromatin typically consists of repetitive satellite DNAs, okay, in the centromere. However, the formation of heterochromatin is not rigorously defined by when a gene is. So let's talk about something called position effect. Okay, this actually talks about if there's anything like gene insertion or translocation, inversion, like anything happens and a gene happens to find itself within a heterochromatic region, it has a higher chance of being inactivated because of its surroundings. Okay, this can be like a genetic let me not spoil myself. So let me just read this for you. When a gene is transferred either by chromosomal translocation or transfection and integration into a position adjacent to heterochromatin, it may become inactive as a result of its new location. Okay, implying that it's become heterochromatic. So based on the surroundings, even if the gene you are taking in is actually achromatic, because of the surroundings, if that surrounding is heterochromatin, it has a higher chance, or it is at risk of being repressed because of its surroundings, and this phenomena is called position effect, right? So this is also epigenetic effect, because in its natural state it is achromatic, okay, but because of its surroundings it has now been inactivated, right? So it's also as a result of epigenetic effect. So the reason why your inserted gene can be inactivated by its surroundings is because the heterochromatic regions can spread. Okay, so you can see this region, the green part is a heterochromatic region, and this part is the active part, the achromatic regions, and you can see the heterochromatin spreading and spreading and spreading. Okay, so your active gene can be inactivated as the heterochromatin surrounding regions spread to those regions. So probably there should be some structures that can prevent these heterochromatic regions from spreading to the active places. Okay, and there is, and we'll talk about it. So let me see if I've missed anything here. So therefore, cells descending from an ancestor in which the gene was inactivated from patches, they form patches corresponding to this. Is not important. Blah, 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 blah. This is because formation of so the closer a gene lies to heterochromatin, the higher the probability that it will be inactivated. I think everything I've said is just to explain this point. So, and this is because the formation of heterochromatin is typically a two-stage process. The initial phase, which the initial stage, which is the nucleation event, occurring as which is triggered by the binding of a protein that will recognize the DNA sequence or other identifiers in the region. So after the protein binds and recognizes the DNA sequence, then the inactive or the heterochromatic region is going to spread along that chromatin stretch, right? Sorry. So the distance by which the inactive structure is going to extend is not precisely determined, and it can be random, okay, and be influenced by numerous parameters or factors like the quantities of limiting protein components and also activation of promoters in the region. If an active promoter may inhibit spreading. So if those genes have like a common promoter, or let's say if there is any active promoter in the region, promoters actually enhance gene transcription. Okay, so if there's an active promoter in the region, then spreading will be limited. Okay, So therefore, genes near heterochromatic this is so as I said, there should be like some structures between these heterochromatic regions and the active regions to prevent them or at least limit their spreading. Okay, so this we call them insulators or barrier insulators, and these insulators can protect and these active regions by preventing the heterochromatic regions from spreading. Okay, so this is what it might look like. Okay, and the green ones are the active places and the orange ones are the heterochromatin. It's actually a protein, so a barrier insulator could bind to the acetyl transferases, leading to hyperacetylation of adjacent histones. You know that acetylation leads to activation of a gene, okay, or chromatin. Okay, so let's look at position effect variegation. So whatever we just discussed was called position effect. Okay, so the ability of your position or your surroundings to cause you to become heterochromatin. Okay, so let's apply this in Drosophila. Like, I don't know, it's a fruit fly. I don't know its name, like its trivial name, but we call it Drosophila. And I said, position effect may differ between individual cells in an organism, and this is called the phenomenon is called position effect variegation. Okay, so genes affected by, so I think variegation, I've forgotten. Variegation is just like changing something that's changing. I've forgotten. But it's actually building a point on this position effect thing. Okay, so as I explained further, you realize there's no anything take here. So genes affected by PEV have two states. Either they are active or silenced, depending on their position relative to the boundary of heterochromatin, which can lead to the variegated phenotype. So variegation, I think, simply means like variation or differences in phenotypes. Okay, as a result of position effect. Okay, so position effect can cause variegation, like simple as that. So we are going to use an example. Okay, using the eyes of Drosophila. Some will have like red eyes, some will have like patches in their red eyes, some also have like full white eyes or black white. Okay, so in this example, some regions in their eye they lack color, right, whereas others are red. Normally they should be like fully red, and because of this position effect variegation, the heterochromatic regions are spreading to the red parts. So some regions will be colorless, and most of the colorless will show as white regions. Okay, so this occurs because the gene that codes for the red pigment is called the white gene. Okay, so the gene needed to cause the red eye is inactivated by position effect. Okay, so the cells in the white gene are going to be inactive, and there will be white instead of red. Okay, whereas the cells in the white gene that are active will give red patches. So those that are inactivated are going to show as white, but those that are not inactivated will still be red. Okay, so the severity of the effect is determined by the closeness of the integrated gene to heterochromatic regions. This is simply stating the effect of position effect. Okay, so white gene is near the tip of X chromosome. Okay, so what if let's take this again. White gene, the gene that's responsible for the red color of their eyes, are found at the tip of the chromosome, X chromosomes, right? So the white gene mutant animals that are white gene mutants, they will have white eyes instead of the red color, the red eyes. Okay, I'm sure you get this as well, because the white genes make them have the red eyes. So if the white gene is inactivated, they are going to have white eyes instead of red. Okay. Chromosomal rearrangement. So what if the white gene that's found like at the tip, or well, let's say wherever the white gene is found, that place there is not enough heterochromatic regions around it. So what if there's like chromosomal rearrangement, some mutations that cause like translocation and that stuff, and the white gene finds itself now in the heterochromatic region. Okay, this can also cause um, inactivation of the white gene. Okay, so mostly this happens in the mutant flies most, and these will cause like the white patches, and some will even have like I'm not sure if they can have fully white eyes, but dominantly white eyes. So an inversion of the piece of the X chromosome will carry the white genes, making sure like positioning them closer to the heterochromatic region. So it's like just I think there should be a diagram. I'm talking plain. So this is your white gene. Okay. And now mutation occurred and the white gene has been swapped, like it's called chromosomal rearrangement. So it is now closer to the heterochromatic region than its initial state. So in its initial state, you realize the white gene is being expressed. That's the plus on it. Okay. But when the ends are exchanged, now it is closer to the heterochromatic regions. So it will now be negative, and the heterochromatic region will start spreading. Okay. So initially, when the white genes are expressed, they will have red eyes. But when the white genes are not expressed, they will have white eyes. Okay. In some cells, the inversion. I've mentioned all this. So I think is this in summary? No, it's not in summary. Expression of a gene can be expressed by virtue of its position in the chromosome rather than by a mutation. So it is not always that. So I think we are just expanding on epigenetics. Okay, so you can have like a genetic disorder. So you shouldn't rush to conclusion that there's been a mutation in the DNA sequence. It can be a change or like a modification in your gene expression. Okay, whether the gene has been activated or inactivated. So this point is simply saying expression of a particular gene can be repressed by virtue of its position in the chromosome. So probably that mutation didn't change your DNA sequence, it just changed the position of the chromosome, and now it has been inactivated or repressed. Okay, so epigenetic silencing can be inherited from one cell generation to the next. I think Doc actually said something in class, differentiating between repression and silencing. So silencing and repression. If I remember, I think repression actually deals with the presence of protein factors like binding to regions. I'm not sure. Yes, you guys should cross check or make sure you search up the difference between repression and silencing. One involves the presence of factors like protein factors, just like transcription factors bind to promoter regions. One of them requires protein factors to bind to specific regions to cause that effect, and the other does not require protein factors. So search for the difference between repression and silencing. In Drosophila and yeast, many active genes are silenced in mosaic fashion when they are relocated to neighborhoods that are heterochromatic. We are still on position effect. I shouldn't be talking about this. We've already mentioned all this. So this part, you have to pay attention. It can be quite tricky. This SUVA and EVA genes. Okay. So the 28 genes in Drosophila affect PEV. Okay, including SUVA and EVA genes. You can call them suppressor variegation genes and enhancer variegation genes. But these names were given to them based on the behavior on the mutant organisms. So if you hear let's take the definition for or what the SUVA genes do and EVA genes do before I make my statement. SUVA genes code for proteins that suppress variegation. Okay. EVA genes code for proteins that enhance variegation. It might look simpler because E means they enhance variegation, SU means they suppress, right? But these E and SU the names were given to them not because in their natural state this is what they do. In their natural in the mutant state this is what they do. So the names were given to them for the behavior of the mutant loci. Okay, so naturally, SU genes will produce proteins that enhance variegation, and E genes will produce proteins that suppress variegation. But in the mutant ones, SU will suppress instead of enhancing, and E will enhance instead of E will suppress instead of suppressing. I don't know if this makes sense. It was a bit confusing at first, but so SU suppressing variegation is talking about the mutant organism, and E suppressing variegation is also talking about, but the normal SU gene does not suppress, it enhances. Okay, so therefore the SU mutations lie in genes whose products are needed for blah 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 blah. So let's talk. I think here, yes. So SU mutations lie in genes whose products. So when we are talking about SU mutations, they are found in genes whose products are needed for the formation of heterochromatin. Okay, because I said the normal SU genes, they enhance variegation. What is variegation? Variegation is the variegation deals with the spread of, like, the position effect variegation, the spread of heterochromatin regions into active site regions. Okay, so the normal SU regions are needed for the formation of heterochromatin. All right. let me take this again. Okay, I've already established these two, that the SU genes, the products from SU genes that act to suppress variegation, and those I have this in my mind. Whenever I'm supposed to say it out, then I'm fumbling. Charlie, it's confusing, so please pay attention. Okay, and if it's not clear enough, if you are if you are my mate, I don't know when you are seeing this video, but if you can get to me, I can explain to you, or you can just search it online. Okay, so let's take it again. The SU genes, okay, they are products. We know genes produce proteins, like they code for proteins. Okay, so proteins that we get from the SU genes act to suppress variegation. Proteins we get from E genes act to enhance variegation. Suppressing variegation means we are getting rid of heterochromatin, okay, or like undoing gene silencing or activating genes. And enhancing variegation means causing heterochromatin or inactivating genes. Okay, but this effect actually found in mutants. So a normal SU gene is not going to do this. A normal SU gene does the opposite of this. Okay, so these two, that's why this is in red. The genes were named for the behavior of the mutant loci. So if that organism or if that locus is not mutated, the SU in its not natural state is going to enhance variegation instead of suppressing variegation. So it is the mutated form that suppresses variegation, not the normal form. Okay, so this the last point here is saying that the mutations lie in genes whose products are needed for formation of heterochromatin. So normal genes that cause formation of heterochromatin. Okay, so if SU mutation come in, then they are going to suppress variegation. Suppressing variegation means they are going to reduce the formation of heterochromatin. Okay, so if the mutation has not occurred in their normal state, they are enhancing the formation of heterochromatin, okay, instead of suppressing it. Okay, and these proteins include histone deacetylases, and we know removing acetyl groups causes the formation of heterochromatin. Okay, and also proteins that are localized to heterochromatin. Similarly, that's how the EVA is. A normal EVA is going to suppress variegation. Okay, that means a normal eva, how do we call it? Yes, a normal eva is going to promote the oh, I'm getting my this thing twisted. A normal eva is going to promote the formation of e-chromatin regions, or is going to activate genes, right? But the mutated eva genes enhance variegation. That means they are going to enhance heterochromatin formation. Okay, so the eva mutations are found in genes whose products or whose proteins are needed in activating gene expression, okay, or undoing heterochromatin formation. And these proteins are members of the SWI-SNF chromatin complex. Okay, so therefore the SUVA genes mutant product. So you see, it's confusing. Into oka one and not two, no oka one. So if you do not understand this, you are going to get them mixed up. Okay. So now we are talking about the mutant product. The SUVA mutant product now will suppress variegation. Okay, because in their normal state, their products were forming heterochromatin, but now there's been mutations in those genes that were forming heterochromatin. So now what are they going to do? They are going to suppress heterochromatin formation. Okay, so the mutants are now going to suppress heterochromatin formation. Okay, so these mutant products are HP1. So HP1 is an SUVA allele or gene. Histone H3 methyltransferase creates methylation on K9, allowing the HP1 to bind. As we said, methylation mostly creates binding sites. Okay, so methylation on lysine 9 is going to create a binding site for HP1 to bind. Okay, in the in the EVA genes, as I said, the mutated ones, those ones too, they are going to enhance variegation. Normally they are activating gene expression. Okay, but because they are mutated, they are now enhancing the formation of heterochromatin instead of e-chromatin, or they are now promoting inactivation instead of activating. Okay, so the mutant genes, and they include members of the SWI-SNF chromatin. Okay, so let's look at telomeric silencing in yeast. Okay, this I had plans of doing this particular model in 3 parts because it's 150 something. It has not looked, it might be more than that. Let's look at this last thing, then we will do part 2 from the mating type switching. Okay, so let's look at telomeric silencing in yeast. Okay, this event is analogous to the PEV in Drosophila, position effect variegation in Drosophila. Okay, so genes translocated to a telomeric region show the same sort of variable loss of activity. Because the telomeric regions, as I said, just like the centromeres, are permanently heterochromatic. Right. So if the genes are translocated to these regions, they are going to incur loss of activity, or they are going to be inactivated. Okay, so this results from a spreading effect that propagates from the telomeres. Okay, and actually what occurs is the binding of a protein called RAP1. So the telomeric repeat will trigger the nucleation event. Okay, so the RAP1 will bind. We see, I think heterochromatin formation, as we said, involves 2 phases: the nucleation phase and the spreading part. So in this particular context, the nucleation phase is the binding of RAP1 that's going to identify the DNA sequence. Okay, and it will trigger the nucleation event. Okay, which results in the recruitment of other proteins that will make the heterochromatin spread from the telomeric from the telomeric region to these genes that are newly integrated into the region. Okay, so I'll see you in the next one, and we are going to talk about mating type switching in